This video is going to cover how the COVID-19 serology test works. This is a test that uses antigen to detect antibodies in the blood of a patient. Unlike the PCR test or the antigen test, this one is used to see if a person has ever been infected with SARS-CoV-2 because antibodies stay in our system for a very long time. In order to understand how this test works, we have to review when different classes of antibodies are made in response to infections. So if we look at panel A, this is showing SARS-CoV-2 virus going into a person's respiratory system, and then it's stimulating the immune cells to make antibodies. Here the antibodies are being made, and you can see the first kind of antibody that's made are the IgM antibodies, which you can see in the chart here. You see the IgM peaks first, and then later IgG antibodies will be made, and they're more abundant than IgM. And so by telling the difference between IgM antibodies and IgG antibodies, we can actually tell whether somebody has been exposed more recently or less recently to SARS-CoV-2. So let's see how the test actually works. Here is the test pad. You'll put blood from the patient here on this sample pad. And if there is antibody present in that blood represented by this little Y shape, then we'll see a positive test result. The sample will flow up the pad, and it's going to flow past this conjugate area, which has lots of different kinds of conjugated proteins on it. It has the spike protein from the virus here, and so if the antibody is present in the blood, it will stick to that spike protein. And we also have a control probe here, which is an antibody that will stick to this last control stripe in the test and let us know that the test is functioning properly. Both the S protein and the control probe will have some kind of colored bead attached to them so that we can see stripes form as they bind to the test. So here we go. The blood with the antibody in it passes through the conjugate pad. As it does, it will bind to spike proteins, and then it will pass along the test going over 3 different stripes. The first stripe has antibody that detects IgM antibodies. The second stripe has antibody that detects IgG antibodies. And the third stripe has antibodies that detect that control antibody. So if the blood sample has IgM antibodies in it, then those will stick to the first stripe. And if that IgM antibody is specific to the SARS-CoV-2 spike protein, then the colored spike protein will congregate along this first stripe and it will turn red. If the blood sample from the patient contains IgG antibody, shown in green here, then it will stick to the IgG stripe, as we can see here. And we'll see a red line in that middle stripe. And then if the test is working correctly, the control antibody, which is also labeled with colored beads, will flow past that third section, and it will bind to the control antibody detecting antibody and stick there so that we get a third stripe. And so finally, we can imagine all of these different possible results from the test. If we get a control stripe here and an IgM stripe, then we know that it's a very early infection because only the IgM antibody is present. If both IgM and IgG antibody are detected, that would be a middle infection because they both are present, would be somewhere in here. And then if we just see IgG antibody in the control line, that means that it's a late infection because IgM levels have dropped and it's just the IgG antibodies that are left. If we just get a line on the control stripe and no detection of IgM or IgG antibodies, that would be a negative test. And then here are some inconclusive results. If you just get a red stripe on IgM or IgG, or you don't get any stripes at all, then it's inconclusive, right? If you don't see a line on that third control stripe, you really just can't conclude anything from that test, and the test would need to be repeated. So this is how the COVID-19 serology test works. And if you ever have a serology test done, now you can imagine what's going on in the lab.